This is our this is our old cancer center. We just opened up a new cancer center at Emory in the midtown. So it's from here. It's just like a short 6-hour drive, or like about a 1-hour flight. But then you have to battle the traffic. So might as well just drive. And thank you, Jenny, and the HealthTree team for having me. She gave me a huge challenge, but really important to think about. In terms of the genetics of myeloma, is what we're doing science or art? And I'm going to try to convince you. I'm going to try to show you science. Personalizing treatment, is it realistic? Is it not yet, or is it not ever? I vote now, not yet. And then is a treatment a relay race to the next best treatment, or possibly a cure? And I'll at the end of the talk, I have a couple slides trying to address that exact issue. This is what we currently do, and I think you've seen this morning. And what I'm about to show you, in no way am I critical of what we currently do. I'm on some of these papers that I'm about to show. I'm not critical about what we do, but what we do today is not what I'd consider to be personalized medicine. So this is how we make decisions, both in the upfront setting and the relapse setting. We put all of these factors together. We have some risk assessment. So we broadly categorize patients into high risk or standard risk. And we put all of this together and to make the treatment decision. But this is the same slide. I didn't make this slide either. This is the same slide. And we put all that together, but we really ultimately ask the question at this time, is the patient a transplant candidate or not a transplant candidate, or eligible? I think that language is likely going to go away in the future. That language was very important, I don't know, 10 years ago or longer, but I think we're not going to stop thinking about transplant candidate or not. We're going to think about, can the patient get the optimal therapy or not? And so that's the major decision that we make at diagnosis, once we establish that you as a patient need therapy. And then we recommend what's the best therapy out there. I think as Christina said, this is a very reasonable choice to say as the best therapy. And then we show you the same slide again. Then we say, hey, look, this is our best therapy. It's better than older therapies because there's more patients going into remission, more patients having a deep remission. Patients stay in remission longer. It's hard to know if there's going to be a survival benefit, but the survival benefit of this study, we're going to talk about when I come back here in 7 years. And then we go and we look and we do things like this, and we retrospectively, we've done the study. We haven't done personalized therapy. We've said you need therapy, you need your best therapy. And then we go back and look and we see which of the groups has done better. We don't make the decision. This is after we've already made the decision what to treat, not beforehand. And the same thing, again, in no way am I critical of this. I was on this study. We participated in this study. And then the same thing we do with transplant, what we call transplant ineligible. We don't think that for whatever the reason, the patient's age, the comorbidities, what other illnesses the patient has, or performance status, how well you're getting around. And then we make the decision, what we can't give what we consider to be optimal therapy, something like 4-drug therapy. So we have a couple of options for 3-drug therapy. We can either give what we colloquially will call VRD light. So it's the VRD, but a kinder and gentler treatment schedule and dosing, or the combination of DARA-RD. We don't have currently molecular markers or really anything to tell us, should we use VRD light or DRD? How in general, how we make the decision to do VRD light or RVD is sometimes we use risk assessment. There are some people out there who say high risk disease, they must have Velcade or a similar drug. it's not clear to me that that's the case. And so in a large part, the physician, whether it's the myeloma specialist or the community oncologist, says, this is the treatment that I like to give for this situation, and that's the treatment that I give. And then the same thing, this is the Maya study that led to the approval of DARA, Revlimid, and dexamethasone as treatment for patients who were not considered to be eligible for transplant. And then you see clearly that daratumumab, Revlimid, and dexamethasone is better than Revlimid and dexamethasone. It makes a lot of sense. You have 3 really good drugs versus 2 really good drugs. When I say dexamethasone is a really good drug, I mean like it helps myeloma and makes your house clean, but it has problems. And then we go back and do the same thing. And we say every subcategory of patient benefited. And maybe there's a group of patients who have high-risk disease that clearly didn't benefit from this therapy or didn't benefit early on, and we need to think about other things. But we're seeing this data after we've already made the decision. So the question is so that's how we currently do things. Again, I showed you that complicated thing where we think about performance status and risk, but in a large part, we're making a decision. Can you get what we consider to be optimal therapy, or we don't think that's possible, so we have these other therapies? And those are the major decisions that we make. And so we're spending a lot of time, myeloma investigators, trying to figure out, can we truly personalize therapy? And while I was being interviewed in the other room, I know that Dr. Tuckman talked about a story that I'm about to talk about. But the first thing that I want everybody to understand about how we treat myeloma today is on this slide. This comes from my colleague, Dr. Larry Boyes. Larry's a scientist. He's brilliant. He understands myeloma more than really, I think, anybody. And our reality about how we treat myeloma is that in a large part, we're not treating the cancer myeloma. We're treating plasma cells. Again, remember, myeloma is cancer of plasma cells. And so almost all of the therapies we have today, all of the therapies we have today are effective because the cell is a plasma cell. Proteasome inhibitors, IMiDs, Revlimid and pomalidomide, the monoclonal antibodies, all of these drugs, they target plasma cells, and myeloma plasma cells happen to be somewhat more sensitive to that targeting. In contrast, and the only art that I'll show you is on the top is an electron micrograph of a plasma cell, and on the bottom is what we call a Circos plot. And the Circos plot is basically a graphical representation of all the genetic changes that happen inside the myeloma cell. And so the question in the cancer biology is, how can we target those genetic changes? There are some malignancies that are all yang, all genetic changes. For example, there's a cancer called CML, chronic myeloid leukemia. Chronic myeloid leukemia has one mutation, and we have brilliant drugs that targets that one mutation. In myeloma, for the large part, we are all on the plasma cell biology and very little on the cancer biology. And so one of the ways that we are trying to personalize therapy, and this is a story that Dr. Tuckman, again, talked about earlier. He talked really about the clinical, and I'm going to talk about how we got to the clinical part and with science. And this is a paper that Dr. Boyes wrote in 2000. This was in 2011, and he said that what we found is that there is a certain, if we targeted a certain protein, the BCL2, that these data have several clinical implications, such as selecting the right subset of patients. So when we say selecting the right subset of patients, that's personalized medicine. That's where we're trying to go. And then he also said, and then we got to choose the right partners for the drug. So the critical thing is, choosing the right group of patients for a drug like this. And so we started asking the question in the laboratory. And every time a patient had a bone marrow biopsy, a little bit of of those cells went up into Dr. Boyes' lab. And Dr. Boyes and my friends and colleagues, Vikas and Shannon, tested this drug, venetoclax, on each and every one of these patients. And not the myeloma cells that came from the bone marrow biopsy. And in the red are all the cells that died very quickly with the addition of venetoclax, and the black are the cells that didn't die quickly. And what you'll note here is that a lot of these patients had the 1114 translocation. And so we started thinking maybe we should just treat patients who have if they have 1114, we should look at venetoclax. And there was a study, a phase 1 study, and this study was done in all comers. And we opened the study at our site. And what we said is that this is not an all comers drug. This is personalized therapy. And so I remember, I'll just tell you a story. I went to, I don't know if Christina's still in the room, but I went to an investigator meeting in New Orleans in 2013, and it was at 6 a.m. because when you're at the American Society of Hematology and you need to have a meeting, they schedule at 6 a.m. So it was in New Orleans, and I was walking through the French Quarter, and there were people still in the bar, and I'm going to my meeting, and I walked in the room, and everybody is depressed about this study. And I said to them, this drug is a miracle. And they looked at me like I was crazy. And I said, why are you treating all these non-1114 patients? If you treat 1114, then as a single agent, there was 40% of the patients benefited. And that's 40% response rate in relapse refractory myeloma is better than Revlimid, better than Pomalyst, better than Velcade, better than Kyprolis, better than daratumumab, better than isatuximab. 40%. And then if you, I'll go back here. And then if you didn't have 1114, and this is why everybody was depressed, the response rate was 6%. That's depressing. That's not enough. Now, the secret is that there's a small number of patients who don't have 1114 who respond, but it's somewhere around 6%. But you can't, 6% is not something, that's not good enough. And 40% is not good enough, but it's a start. It's a start to personalized therapy. And then again, what we showed in putting all that data together, this is a graph showing that patients with 1114 are more likely to be sensitive to let me rephrase that. Patient cells are more likely to be sensitive. And so this is the patient, this is our patient. She was 38 years old, had refractory disease, and her sister was a go-getter and found this trial and looked up Dr. Boyes' research and said, this is the trial for you. And she immediately responded that this is a graph of her paraprotein. This is 2014. And then this is her hemoglobin. So her hemoglobin got better. So not only did the protein go down, the hemoglobin got better. That means she got better. And this goes like, this is COVID, so she didn't visit us. But this went up until, this is what, like 7 years. And at that moment where the arrow is, she and her sister visited this mountain. All right, an extra cookie for who knows this. Yes, this is Mount Fuji in Japan. So this was from this is the promise of personalized therapy. And then we did this all, and then we looked at many more patients, and now we know that if you're in the green, if the myeloma cells, we do a simple test, we do a bone marrow biopsy, send a tube up to the lab, they take the myeloma cells, they put venetoclax on, and then ask the question, how many cells died and how quick was it? And if it worked really fast, then you're green. And what you'll see is that if you're green in the laboratory, then you're green over here, and those are the patients who are responding. So this is true personalized therapy. Now, the problem is, well, not the problem, but so far, this is our largest, and not quite only, but one of our only true personalized therapies. And then Dr. Gupta Vikas in the lab is because it's not reasonable to do a bone marrow biopsy and send it to a lab and have them do that test. That's never going to be the test. And so he was able to show that if by simple markers on the myeloma cell that can be run in any lab, in any flow cytometry lab, any pathology lab in the country, and we can have a score, and if you're score 1, then you're likely to be sensitive, and if you score 0 or less, then you're not sensitive. So when we talked about the tau of myeloma, and we had the myeloma, what's happening in the genetics, here is a non-exhaustive list of everything that we could possibly do. And there's one really interesting story of we've found that about 5% of relapsed myeloma patients will have what's called a BRAF mutation, V600E mutation. And this is a mutation that's common in other cancers. It was first seen in melanoma, and they developed drugs for melanoma. They've learned that a BRAF inhibitor, in combined with other inhibitors like a MEK inhibitor, is effective in other malignancies. And we brought that to myeloma, and it's clearly a very effective therapy. So this also, in that 5% of patients who have the V600E mutation, this is personalized therapy. And this is it's something that can only be discovered by doing additional testing, things like next generation sequencing or genomic testing, looking specifically for this abnormality. Okay, and then finally, I have a couple slides asking the question: are we in a relay race or a cure? So this is what we currently do, which is the patient had whatever therapy up front and now has relapse disease, and we ask the question, what did you have? Are you progressing on Revlimid? If you're progressing on Revlimid, we have a series of things, and if we do this first, and then if that stops working, we do this next, and if that stops working, then we have this and this as options. And this is what we currently do. And I will argue, and we actually have named the trial. This is called what we call the cure trial, because we just forget about that relay race and getting all the drugs. Let's figure this out. We believe, because when at the initial consult, when you're diagnosed with myeloma, one of the things that every doctor says is that this is not a curable disease. We still say it. We believe, we don't know, we believe that the tools are in place to make this a curable disease. And when you say that in a bunch of myeloma doctors, everybody gets, they say, but we believe it. And so we're going to do this trial, and we're going to get every drug, and a cure means you've had treatment. You've had treatment, you're off treatment, and there is a good chance that you will never need treatment again. And that's what our goal is here. And we're taking all of our best therapies, and we're combining this. It's going to be, this is just our design, and it could be 2 years. And I intentionally took all the names of the drugs that we're going to be putting in here off, because it's not there yet, but we're going to have 3 drug combinations combining with other 3 drug combinations, classic oncology, putting drugs together with non-overlapping toxicity. And then then a CAR T cell. If we don't have a CAR T cell, we're still thinking about transplant, maintenance therapy with bispecific antibodies, MRD testing, and the entire time we're asking the question that to try to understand how this is going. So I believe that this is going to be the future. This is what we do today, and this is what we've done, but I believe something like this is going to be the future. And I think with that, I'm done. And this is all the people that I work with that do the work. And I, again, appreciate you having me and hosting me here. Thank you very much.